I'm in charge of the mosquito control laboratory at QIMR Berghofer, and my background is originally in agricultural entomology, and then I spent several years as a medical entomologist in South America and then East Africa before joining Queensland Health to run their dengue management program before the release of Wolbachia in northern Queensland. I changed my title because I had to look at what else was being spoken about, and I thought it was better to be general than specific. And Hamish has already covered quite a bit of the kind of discussion around Ross River. So I will still talk about Ross River, but it's not the focus anymore. One of the focuses is really that you see an awful lot of shorthand in terms of the relationship between climate and mosquito-borne disease. And a lot of it is based on models which are extremely inaccurate, and it's then picked up and reported by the media. But there's this general idea that increases in temperature, because they may promote the poleward movement of vector species, are then going to have a dramatic impact upon parasites and pathogen transmission. And it's really not the case. As we know, a lot of vector-borne diseases are in decline, despite the very evident impacts of climate and lots of other aspects of ecology. And there's this idea that vectors particularly have these very specific upper and lower thermal optima. And it's just really not particularly valid, especially for things like Aedes aegypti and to a lesser extent Aedes albopictus, which are the main vectors of things like dengue and Zika and chikungunya. But as we'll see, the fact is that things don't really ascribe to specific temperature optimum, and they're perhaps of limited value. It is the arrow keys meeting.
Let's see what's going on here. There we go.
Great. So I think, as we already heard this morning, and there's been a fantastic bunch of talks in terms of the breadth and scope, it is complicated. And so it's not as easy. So everything is from globalization to urbanization to the mitigation of disease through biosecurity, vector control, vaccines and drugs, changes in human behavior, changes in mosquito behavior depending on environment. Then those all interact before we get to the stage of kind of human transmission. So that's a messy cartoon, but it is just, I think, saying what we've all been saying this morning, which is it's complicated. So the most obvious example, perhaps, of where there really is no impact between expected impact between climate and some disease is if you look at malaria in Europe in the early part of the 1900s. Then, of course, we know that climate change is obvious in Europe. We can see that all those hottest years being lumped up after 2000. But then malaria was once an endemic disease in large parts of England and the UK. And that was because it was spread by a particular mosquito that's part of the Mosquito-borne complex that bred in brackish waters around various swamplands. Lots and lots of servicemen returned from the First World War with parasites, and then tremendous circulation of the parasite in the local environment. But things changed very quickly. The military began its own chemoprophylaxis in terms of quinine. The swamps were drained, beef and dairy cattle were brought in. Atroparvus happens to be a mosquito that likes to feed on cattle. So again, this tremendous dilution effect. Clearly the parasites are only in humans, and so any mosquito feeding on something else is not able to transmit anymore. And so increasing climate, but absolutely no increase in malaria. And that would be expected in many places around the world, not least because of increasing urbanization in sub-Saharan Africa. And urban environments are really not good enough for these environments. If you look at a place like Cambodia, they have 0 deaths from malaria now. And that's because they basically built themselves out of an enough needs population. They've got hardly any forest left, and that's where all the transmission needs to occur. The other point about these sort of thermal optima and Aedes aegypti. In 2010, the city of Ahmedabad in Gujarat had a kind of a heat emergency when at one point 300 people died in a single day. The minimum temperature throughout almost the entire month was above 40 degrees. And that's well above the optima that are expected to be conducive to the proliferation of Aedes aegypti. But at the same time, that same year, was one of the worst years in India for dengue. And Gujarat was the state that had the highest incidence. So Aedes is one of those really interesting animals that is so beautifully adapted to the human environment but it exploits it in order to avoid all those challenges of temperature. So in an urban place, it's got a multiplicity of blood meals from the human crowded population. It's got dark, humid environments where it can survive temperatures that it wouldn't survive if it was outside. And it has myriad oviposition sites, of course, because of all the water storage behavior that people do, which is a potential impact of climate on Aedes aegypti. It may be that indirectly human behavior will lead to increased water storage or change water storage habits. And the dengue risk may be increased that way. I mean, it's certainly true that Aedes aegypti, as an example, is, it is still spreading. It's still spreading through California. It's still spreading through large parts of Asia. And it is in every continent, of course. And here's something done by Narayan Jiawali in our lab, just looking at patterns in dengue over the past few years. And you can see that with the very obvious impacts of climate on temperature elevation, then dengue is moving up to those higher elevations where it's now circulating autochthonously in cities which are really quite high, like Kathmandu. And interestingly, Aedes aegypti has only recently invaded Nepal. They had Aedes albopictus for the last century, but Aedes aegypti is a relative newcomer, and that may be another reason for increased dengue transmission in that country. And one of the things that it still surprised me that we hadn't really got to grips with in Queensland is the fact that there is no doubt that Aedes aegypti is coming back at some point. It was here right up into the 1950s, and then something happened, a change in, possibly a change in water storage habits and decline of the mechanisms for watering the rail network, widespread use of insecticides, the DDT, and lots of legislation where the council actually did a pretty good job of making sure that people were not breeding mosquitoes in their backyards. That's all stuff that's sort of now disappeared. But so Aedes aegypti now just exists in the north of Queensland and in a few pockets around central Queensland. And it's a very genetically homogeneous population. It's probably not very adaptable, but the risk doesn't really necessarily come from those little remnant pockets. It comes from the fact that there's this incredible propagular pressure of invasion at Queensland's borders, international ports all the time. So I think it's only a matter of time before it gets here, but we've got no real plan as to what we do when it does. Do we suddenly rear lots of Wolbachia infected Aegypti and release them in Brisbane? Do we have an elimination plan? We have none of that kind of in place, but there are definite and real risks. And they are, to some extent, climate related, because Queensland will increase its temperature and we will go from being temperate to more kind of subtropical in terms of average temperatures. And in terms of the survival of the mosquito, these are experiments in small water containers and larger tanks in Brisbane. And Brendan Truen, who was a PhD student in the lab, just showed that these large water tanks are excellent buffers against Brisbane's colder winter temperatures. And mosquitoes will just hang there as larvae for as long as it takes for the temperatures to increase, and they can develop once more. In buckets, if you can see the dashed lines, temperatures in buckets often kind of go below the supposed lower critical temperatures for Aegypti. So they're not, a mosquito, depending on those environments, will have a tougher time than those in rainwater tanks. And rainwater tanks are something which are expanding rapidly throughout southeast Queensland and elsewhere, partly in relation to the unpredictability of rainfall. So again, an indirect impact between climate and the ability of mosquitoes to proliferate. And this is, I won't dwell on this at all, but it's just making the point that mosquitoes will hang there in their buffered environments for as long as it takes. So taking 36 to 40 days to hatch is an incredibly long time for mosquito like Aedes aegypti. But you can imagine that just an increase of 1.5 degrees or something in Queensland will very rapidly increase that development time and make it easier for mosquitoes to proliferate. We just talked about Ross River briefly. Hamish talked about it before. Amanda Murphy, who worked with Al Skinner on some aspects of the Ross River investigation, did a spatial analysis of Ross River in southeast Queensland and found the black dots in the north are the bigger hotspots and the permanent hotspots for Ross River. And you can't really see the detail in this map, but it is people were talking about this morning, it occurs at the edge. It's where the urban environment is in transition towards the kind of peri-urban or rural environment. So again, these edge effects and potential impacts on diversity are obviously some sort of driver of transmission. Interestingly, the little red promontory sticking out right in the kind of middle of that map, we recently did one of the biggest SERA surveys of marsupials that's ever been done. We did it on a whole bunch of koalas that were tagged and bled regularly as part of the Moreton Bay Rail project. And interestingly, about 80% of them had seroconverted from Ross River, and they seroconvert very quickly, within the first year of life. Where it's interesting that little promontory that you can't really see there, I don't know if I can point to it, it doesn't have any hotspots associated with it. So there's obviously something, there are things which are not well explained in terms of, why are all the koalas seroconverting, but there don't seem to be a large number of like human cases. And then Hamish was talking about the human transmission of Ross River, and the underlying, the red lines there are just saying that the humans are definitely one of the suspects, but they've been a suspect for a long time, ever since the late 70s, with the big outbreaks in the Pacific. We knew that there was human to mosquitoes, a human transmission going on. And the little graph there is what we did with Colleen, just showing that in Fiji, almost all domestic animals are seroconverting. So there's something going on in the absence of a mosquito kind of host. The last thing I just want to say is we need to do more around surveillance. What we have is not adequate. We gave up, for example, on the Japanese encephalitis surveillance quite a long time ago. And all we're left with is a rather clumsy kind of mosquito surveillance and sequencing exercise, which is very bad at predicting kind of what's out there. So I think one of the things in terms of thinking about infectious disease and how we monitor it is we have to increase our capacity to do something better around surveillance and ultimately around control as well, because it's one thing to describe a problem, it's quite another, especially for some of these complex zoonoses, to actually find a vulnerability that we can focus on. Thank you.